in any of the work that you've done, have you seen any correlations with differences in environmental qualities that might influence CBC in any sort of way? And the reason I ask is there's some old literature. I mean, possibly, I can't remember when it came out. It was possibly late 90s or early 2000s, so it could be totally irrelevant now. But it was based on looking at some of the data that was coming out of the University of Mississippi and some of the accessions that they had. They were seeing higher levels of CBC, and there was speculation that it might have something to do with humidity or something like that, given that the plants were grown in Mississippi versus, more arid environments where they normally would be grown.
So it's a really good point. And one thing that surprised us in this whole study is after we sequenced these 40 genomes, and there's a figure in the paper I should point people to in this that may give them a perspective on it. Let's see which one it is. I think it's figures 7, 8, or 9. What we looked at was the presence of these copy number variations. Your audience probably can't see this, but what shocked us, we'll maybe post it in show notes or something, is that the blue means they're deleted and red means that you have the genes. The Jamaican lion strains are here purple. They're heterozygous for it. But what was shocking to us is that there are all of these different strains out there that don't have this cannabichromine cluster. And this isn't one gene. This is 2 million bases of sequence that's deleted.
Wow.
And what's on that contig that's been deleted, or the portion of the contig that's deleted, happens to be 3 or 4 other genes that are involved in pathogen response. There's an aquaporin gene in there. There's a few of them listed in the paper, but so if there's any cannabinoid that would be most directly linked to environmental conditions and perhaps pathogen response or humidity, I would pin it on that one, because you can see that there's a common deletion in the population that when it's gone, you lose some of these other genes with those cannabinoid synthase genes. And so those plants arguably are either going to be more prone to pathogens or have a different response to stress one of them is actually a gibberellin transporter, right? That gibberellin is a really important plant hormone, and I don't know how many more the genome has. I've got to go and look at that, but just seeing one of those things get deleted means the plant's probably going to have a really odd response to stress, and it's in very close proximity to the cannabichromine genes that might be reason to believe that the cannabichromine expression might be a little bit more linked to these types of changes.